apoptosis is a form of programmed cell death during which the cell doesn't explode as a necrosis, but splits up into closed and organized fragments called apoptotic bodies. Apoptosis, which can be either physiological or pathological, is necessary to maintain homeostasis in different tissues. Moreover, apoptosis is a form of inflammatory death. In fact, not only is it a prevention of inflammatory phenomena of necrosis, but also leads to molecule production suppressing inflammation. Physiological apoptosis can be observed in embryogenesis, during which its role is to destroy structures that don't have to be present in near-term fetus, as for example, membrane between fingers of hands. Another form of physiological apoptosis is the hormone-dependent evolution that can be seen in menstrual cycle. That is to say, the atrophy of endometrium at the end of menstrual cycle depends on cell apoptosis induced by hormones. Finally, apoptosis plays an important role in maintaining homeostasis in the immune system, since it removes cells that don't work properly. Examples of pathological apoptosis are instead those occurring after DNA damage. In this case, apoptosis is given through particular proteins, also called checkpoints, which induce the cell death in case of problems in cell cycle. This phenomenon stops the transmission of gene damage to other cell generations. Another example of pathological apoptosis is virus-induced apoptosis, as for HIV, hepatitis B and C. Finally, apoptosis from misfolded protein accumulation is a phenomenon exposing to stress the endoplasmatic reticulum that completely stops cell function. In this case, the cell cannot be saved through other systems and, being harmful, it has to be eliminated. From a morphological point of view, with apoptosis, cell decreases in volume. As in necrosis, protein denaturation leads here to significant cytoplasmatic eosinophilia. Contrary to necrosis, in apoptosis, protein denaturation is caused by enzymes that activate during apoptosis. At the same time, the nucleus, which is now fragmented, enters karyolysis in a controlled way, and the DNA is cut into fragments of equal length. However, the most interesting event is undoubtedly that of apoptotic body formation. Cytoplasm doesn't spread over, but is gathered in membrane vesicles that are then phagocytized by macrophages. Consider that apoptosis was named after apoptotic body formation. In fact, its name means fallen leaves because apoptotic bodies detaching from cell body remind of autumn leaves falling from a tree. Under the optical microscope, we can see that the apoptotic cell tends to be much more eosinophil but remains gathered and doesn't spread. We can also observe karyolysis. Moreover, apoptosis is biologically mediated by very important enzymes called caspases, that is to say, cysteine proteases cutting a residue of aspartic acid. All caspases have an inhibitory domain to be cut in order to activate them. Sometimes this domain blocks the active site of enzyme only. Other times, its cut rearranges molecules, leading to active site formation. Caspases can be divided into initiator caspases and effector caspases. Initiator caspases 8 and 9 are those beginning the apoptotic pathways that we are going to see in a moment, while effector caspases are activated by the initiator caspases and lead to the activation of the enzymes, destroying the cell in an organized way, starting from the intrinsic pathway. Apoptosis. The intrinsic pathway, which may start from mitochondria or from the nucleus, is a form of cell death occurring when cells cannot repair damage. So, the intrinsic pathway is actually called intrinsic due to the fact that it comes from the inside. Mitochondrial intrinsic pathway has been studied for many years. As previously stated, when talking about mitochondrial damage, there is a channel protein of mitochondrial wall which is normally closed. This channel protein is called mitochondrial permeability transition pore. When opened, the pore connects the mitochondrial matrix with the cytoplasm. This closing mechanism is artfully regulated. The mitochondrial permeability transition pore is composed of two proteins, a protein on the external mitochondrial membrane, the VDAC or voltage-dependent anion channel, in a protein on the internal mitochondrial membrane called ANT or adenine nucleotide translocase. During radical-dependent cell damage with increased calcium ions or protein folding, the opening of the mitochondrial permeability transition pore is stimulated. This stimulation occurs through the interaction with cyclophilin D, a molecule located in the matrix. There is therefore an exchange between the matrix and the cytoplasm. The calcium in the mitochondrial matrix is released in the cytoplasm. Vice versa, the matrix absorbs water from the cytoplasm, and so the mitochondrion swells. The mitochondrion has to support an osmotic stress that is higher than normal, and for this reason, the water influx causes mitochondrial explosion, and the matrix substance outflow is even more significant. However, this phenomenon preludes cell necrosis and not apoptosis. The mitochondrial permeability transition pore was long thought to be involved in the apoptotic process via the intrinsic pathway. But the apoptotic process via mitochondrial intrinsic pathway occurs only with DNA damage, growth, signal reduction, or protein misfolding. In this type of damage, only the external part opens up, the VDAC, allowing the outflow of two proteins stimulating apoptosis, cytochrome C and Diablo. It was impossible to understand that the mitochondrial permeability transition pore was not completely involved in programmed cell death. Only its external portion was. It was found that, contrary to necrosis, substances go out from the intermembrane space and not from the matrix. It was then clear that mitochondrial permeability transition pore is regulated by a family protein, the so-called BCL family protein, or B-cell lymphoma. The most common is BCL2, a negative regulator of VDAC. In fact, BCL2 binds VDAC and induces its closure. Besides BCL2, other family proteins were discovered, as MCL1 and BCLXL. MCL1 and BCLXL were named BCL anti-apoptotic, since it was then found that there are other proteins of the same family that can open the external part of the mitochondrial pore to favor the outflow of pro-apoptotic molecules. Among the others that are BAK, or BCL2 antagonist killer, and BAX, or BCL2 associated X protein, which were named BCL pro-apoptotic proteins. Later on, it was actually discovered that the main role of BAX and BAK is that of inserting themselves in the membrane, forming homodimers and heterodimers to open the channels. It is important to outflow cytochrome C and Diablo through BAX and BAK than their regulatory action on the pore. However, since they are both pro-apoptotic and anti-apoptotic molecules in the same family, the point is, how are proteins regulated? Finding the regulatory proteins of the BCL family was therefore extremely important. BAD, BIM, and BID are BCL that stands the type of cell damage. BID is the most studied because of its ability to suppress anti-apoptotic molecules and activate the pro-apoptotic ones. In fact, it stimulates the heterodimers and homodimers of BAX and BAK to insert in the wall. All this explains what the possible pathways of cytochrome C translocation into cytoplasm are. But the counter-regulatory functions among BCL proteins are not over. In fact, BCL2 and other anti-apoptotic proteins directly inhibit BAX and BAK. At the same time, they can inhibit the anti-apoptotic protein of BCL family. This mutual inhibition usually maintains some kind of balance inside the cell. However, in case of damage, BID protein blocks the anti-apoptotic proteins and stimulates the pro-apoptotic ones. And this causes an imbalanced counter-regulatory function while favoring channel insertion and allowing the continuation of the apoptotic mechanism. At this point, cytochrome C, leaked out from mitochondria, flows into the cytoplasm where it meets a molecule called apoptotic protease activating factor 1. Cytochrome C is normally bonded with BCL2 molecules, but when pro-apoptotic situation occurs, pro-apoptotic molecules block BCL2 molecules, detaching them from APAF while inducing a bond with cytochrome C and conformational exchange of the molecule. Hence, the molecule reveals its domains. There is a domain called WD4A binding cytochrome C, a series of CED4-like domains forming a sort of chain, and finally a caspase recruitment domain or CARD. Since the molecule activates initiator caspases through it, CARD is an extremely important domain in cell transduction. Initiator caspases are normally controlled by a protein called inhibitor of apoptosis protein, or IAP. IAP is detached from the initiator caspase, which in the case of the intrinsic pathway is caspase 9 by means of Diablo, direct IAP binding protein with LOWPL. At this point, the cytochrome C bonded with APAF can form an aptamer with other identical molecules. And this is in order to reach the maximum affinity for caspase 9. So, a body with 7 cytochrome C, each binding its APAF and a caspase 9 will form. This structure is called apoptosome and isn't active yet, and this is since caspase 9 is still maintaining the inhibitory domain. At this point, CARD detaches the inhibitory domain of caspase 9, leading to an active form of apoptosome inside which it cleaves and therefore cuts and activates both other caspases 9 and effector caspases, among which caspase 3. Intrinsic pathway from nucleus is completely different. It is triggered by a DNA single-strand break that attracts a repair enzyme called poly-ATP ribose polymerase PARP or PARP1. When this enzyme binds a single-strand break, it reveals its site and attracts ADP. It then attaches a molecule of ribose to ADP, forming an ADP ribose and polymerizes identical monomers, forming a poly-ADP ribose. Chain formation has only to waste the ADP inside the cell so that it won't be used to continue the DNA synthesis until the strands are repaired. If strand repair takes too long or is impossible, a poly-ADP ribose will succeed in going out of the nucleus. Its interaction with calpain is known. In fact, this interaction frees calpain from its inhibitory domain and leads it up to the mitochondrion, where it activates BID. Since BID action is pro-apoptotic, it induces BAX insertion into the external membrane. Moreover, through a mechanism that is still unknown, calpain induces the apoptosis-inducing factor activation of the intermembrane space. The apoptosis-inducing factor then translocates into the nucleus and, through an uncertain mechanism, leads to the nucleus segmentation and fragmentation. The extrinsic apoptosis pathway differs from the intrinsic one because, as the name suggests, it comes from the outside. In this case, apoptosis is triggered by a series of molecules binding cell surface receptors, inducing cell death. The receptors involved in this phenomenon are type trimeric or TNF. That are mainly Fas receptor or CD95 and TNF receptor or TNF-R1. When bonding their ligand, which for Fas receptor is Fas2 ligand, death domains interact with each other by attracting a molecule called Fas-associated protein with death domain, or FAD-D. After caspase 8 is free from its C-flip inhibitor, through some unknown mechanisms, FAD-D, with its own executive death domain, attracts caspase 8. The interaction between FAD-D and caspase 8 leads to caspase 8 detachment from the inhibitory domain to its activation. TNF receptor 1 pathway is instead more complex since it induces a high number of responses. The adapter here is named TRAD or TNF-associated protein with death domain. TRAD-D may attract a lot of molecules with different effects, among which RIPK1 or TRAF2. TRAF2 may even activate apoptosis inhibitors. However, sometimes TRAD binds FAD-D and FAD-D, and precisely like the mechanism of Fas receptor activates caspase 8. When caspases 8 are active, they tend to multimerize with each other in order to increase their activity. Later, they cleave caspase 6 and caspase 3, leading to the activation of the effectors. In this case, there's no real apoptosome formation. Apparently, the two pathways we've seen until now are not so different. Caspase 8 can induce BID, which induces BIX and other BCLS pro-apoptotic protein translocation by stimulating the outflow of cytochrome. The two pathways are probably not completely closed, but the activation of one minimally activates the other one, and in addition, they cooperate to induce the effector phase. The effector phase of apoptosis is in fact identical. Effector caspases 3, 6, and 7 mainly activate proteases which cut cytoplasmatic proteins. DNase then cuts the DNA and activates a number of molecules inducing apoptotic body formation. Enzymes cutting the DNA have a specific role. In fact, caspase-activated DNase, or CAD, cuts the DNA into fragments of 50 to 300,000 base pairs. These base pairs will be cut into fragments of 100 to 200 base pairs by an endonucleases activated by the caspases. As for the apoptotic body formation, little is known about mediating enzymes, but what we know are some specific processes making apoptosis an extremely organized death. Ketone bodies do their best to be eliminated with the least possible inflammatory effect. For example, phosphatidylserine that is present in the inner layer of the membrane is brought to the external layer through the enzymes called flipases. Phosphatidylserine is very important for phagocytosis, since macrophages phagocyte everything with externally exposed high concentrations of phosphatidylserine. Moreover, it is believed that apoptotic bodies can produce chemoprotective factors for phagocytes, especially for macrophages. Apoptotic bodies tend to cover fragments of complement and natural immunoglobulins, opsonins that mediate phagocytosis, especially by macrophages, not for immune system related purposes but for debris elimination. In fact, the macrophages may act like an antigen presenting cell, phagocytizing and processing cell fragments and then presenting them to C lymphocytes. But in this case, there is a risk of cell reactivity. Apoptotic bodies can induce phagocytosis alone without any immune system processing. So, as we saw earlier, the main causes of apoptosis are the lack of growth factors, since receptors of growth factors activate anti-apoptotic BCL, then DNA damage, since damaged sensor proteins can activate pro-apoptotic BCL, and misfolded protein accumulation. The exact mechanism inducing apoptosis isn't clear yet, but we do know that apoptosis plays a remarkable role in neurodegenerative diseases and that apoptosis is essential for the immune system. First of all, we gotta say that fast ligand-induced apoptosis is used to maintain tolerance to the self molecule, that is to say, to kill self-reactive lymphocytes. In addition, cytotoxic T, CD8 lymphocytes use granzyme B as a killing mechanism, which is translocated into target cells. Granzyme B then activates procaspase 3 by cleavage, converting it into active caspase 3, which will then induce apoptosis in the target cell. In a nutshell, the difference between apoptosis and necrosis is crystal clear. Apoptosis is an extremely organized form of cell death, while necrosis is extremely messy and inflammatory. In both cases, there is a cytoplasmatic eosinophilia increase due to protein denaturation that, in case of apoptosis, is precise and mediated by enzymes, while in the case of necrosis, is imprecise and damage mediated. In apoptosis, there is apoptotic body formation, whereby in necrosis, membrane rupture and cytoplasm outflow, as well as myelin figure formation mediated by lipid damage occur. In apoptosis, the nucleus is cut into microfragments of nucleic acids of 180 to 200 pair bases, while in necrosis, the rupture is faulty and extremely irregular. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.